Another group of alveolates, third group of alveolates, and the last one that we'll talk about today are ciliates. So ciliates have cilia. It's very, very easy. So cilia, if you don't remember what those are, you might have to go back to Bio 181, those little structural projections of the plasma membrane that are used for various different things. In this group of organisms, they're used both as movement and to feed. So for example, the little cilia on the edge of the Vorticella cup are for feeding. And most of them are predators, and they prey on bacteria or other small protists. What's really cool about ciliates is that they have two nuclei, a big one called a macronucleus and a smaller one called a micronucleus. The reason they have this, the function of this, is for this really weird sexual process, horizontal gene transfer, that is called conjugation, and that we will look at in a moment. So one of the most common of the ciliates, one that we usually get to look at in the lab, and so again, this is where it would be great to look at the photographic atlas and look up some of the ciliates, is Paramecium caudatum. Okay, so this is the genus, and there are multiple different species. And it is, if you look at all of its bits and parts, it is like a miniature organism. It doesn't have a mouth, but it has a mouth pore, okay, a mouth-like device. It's covered in these little cilia, and the cilia kind of bring the bacteria towards this mouth pore. And the mouth pore then, guides this food, if you will, towards these food vacuoles, which once digestion takes place, these vacuoles then, join with the plasma membrane to form an anal pore. Okay, it's reminiscent of what animals have. Okay, they also have a contractile vacuole. Aha, what's a contractile vacuole? What does it do? Does anybody remember that? I'll give you a hint. It has to do with the fact that Paramecium lives in fresh water. It helps get rid of water. Yeah, that's exactly right, Rebecca, because the environment inside of this organism is hypertonic, right? Just like the inside of our bodies. Think about your saliva, your tears, your blood, etc. It's hypertonic, meaning that it is saltier relative to what's on the outside. What's on the outside, if it's fresh water, is hypotonic. These organisms don't have a cell wall, which means they're continuously taking on water, right? Water goes from where it's in greater concentration out here, it's watery out here. So water goes from where there's more water to where there's less water on the inside. And if these organisms didn't expel this extra water, if they didn't squirt it back out, okay, if they didn't, I don't know how to represent a little splash, but if they didn't squirt it back outside of the body, they would lyse. The cell would break. It would lyse. Okay, so this is something that the cell has to expend energy to do, but that's the function of that contractile vacuole. And it's not spelled vacuole. This is misspelled. It should be vacuole. Okay. All right. So let's talk about this business of having a macronucleus and a micronucleus. It is for this special process of conjugation. Let me see if I can find some older notes that kind of explain this, because I have a diagram to show you, but if I can diagram it right from the start for you. Let me see. and that might help. Okay, so it's been a year since I've done this, so bear with me. I didn't practice this today. But basically, this is how conjugation works. Okay, so this is in Plasmodium. So what I'm about to show you is conjugation in Plasmodium. Okay, and it's a form of horizontal gene transfer. This life cycle, the life cycle of this organism, the sexual life cycle, involves meiosis, mitosis, and binary fission. It's kind of crazy. Okay, so it takes two. Okay, sexual reproduction, a type of sexual reproduction always takes two different individuals. And there's a macronucleus, and each one has a micronucleus. All right, let me get a different colored pen to show you that they they're different mating types. Okay, so this is a diploid micronucleus. It's diploid and it's a micronucleus. It undergoes meiosis. Okay? It undergoes, this micronucleus undergoes meiosis, but no cytokinesis. Okay, so because it undergoes meiosis, the end result is you still have this macronucleus. It's 4 micronuclei, but now they're haploid. Okay, these are N haploid micronuclei. You guys with me so far? Because, okay, this guy underwent, let's see, meiosis 1. This is meiosis 1, produced 2 daughter cells, and meiosis 2, 4 daughter cells. Okay, that's how we went from 1 to 4. 4 of these actually disintegrate, and nothing happens to them. Okay, this one undergoes mitosis. So what does it look like? Now you have this one cell with a macronucleus and a haploid, or I'm sorry, 2 haploid micronuclei. Okay, so again, mitosis, no cytokinesis. No cytokinesis. So the rest of the cell as a whole doesn't divide. It's only the nucleus that divides, only that micronucleus that divides. Okay. Meanwhile, the same thing is happening down here in our other mating type. Okay. So we have this diploid macronucleus, and it undergoes meiosis with no cytokinesis. So we've got a macronucleus, and then I'm gonna go ahead and, so I don't have to. And then it produces, and actually I'm gonna draw this one up here, because conjugation, they have to physically be close to each other for this to happen. Okay, so this produces 4 daughter cells, 1, 2, 3, 4. And then we've got 2 micronuclei here, and these are each haploid. Okay, these are haploid, and 3 of these disintegrate. Okay, so it's just this one right here that undergoes mitosis to produce these 2 right here. Okay, during conjugation, okay, this paramecia, my hairy little, they've got these little cilia on them. This thing happens, this happens. One of these goes here and one of these goes here. Okay, so after conjugation, what you end up with are two cells that look like this. They have a macronucleus, cilia, and then they have one of each micronuclei. Ooh, look at that. Okay. And then what do you think happens? Nuclear fusion. It's not just for physics. So you still have your macronucleus, but these guys fuse together, right? So you've got this is haploid, this is haploid. So what do you end up with? You end up with a diploid micronucleus. This diploid micronucleus undergoes 4 rounds of mitosis. 4 rounds of mitosis. So you end up with 1, 2, 3. No, 3 rounds of mitosis. I'm sorry. So this undergoes mitosis 3 times. So this becomes this. Okay. This original macronucleus actually disintegrates. And then because you've gone through 3 rounds of mitosis, 1 becomes 2, 2 becomes 4, 1, 2, 3, 4. And then in the 3rd round, you end up with 8 of these. Four of them become macronuclei. 4 of these after the 3 rounds of mitosis. 1, 2, 3, 4 of them become macronuclei. 4 of them remain micronuclei. We're still all inside of one cell. It's kind of insane. This cell then undergoes binary fission, meaning that the cell actually splits, and it undergoes 2 rounds of binary fission such that in the end you end up with 4 cells, each with a macronucleus and a micronucleus, and the cycle starts over again. Did y'all follow that? Because I was so focused on the board, I did not pay attention to anybody's faces or expressions or anything like that. Okay, so this is my diagram that I drew, years ago that I had to practice, because when I looked at the diagram in the textbook, I was like, holy cow, how on earth, what is going on here, right? And so you kind of have to call to mind and remember, okay, what is mitosis? How does it work? How many daughter cells does it produce? Does it change the ploidy of the cell? No, it doesn't. It's the photocopy process. Okay. What is cytokinesis? Okay. Mitosis and meiosis, for that matter, is just division of the nuclear material. Cytokinesis, division of the cell. Okay. Binary fission is division of the cell. So once we've gone through all this, or maybe you want to go in a reverse direction, when you look at this, it's hopefully not going to make your eyes go all cross-eyed. Okay. So let's see. Where do you start here? Well, I guess it would make sense to start with number 1. Yeah. It's pretty much what I just showed you. Okay. Meiosis produces 4 haploid micronuclei, 3 of which disintegrate. You've got 2 remaining. They swap places. Then they fuse. Okay, a single A diploid micronucleus. Okay, these are haploid at this point. These are all haploid. They undergo 3 rounds of mitosis to produce 8 micronuclei. The original macronucleus disintegrates. 4 of the micronuclei become macronuclei, right? 4 are micronuclei, and then 2 rounds of cell divisions produce 4 daughter cells, each of which has 1 macronucleus and 1 micronucleus.